You're going to be recorded twice, Cheryl.
Right, I agree.
I think it's, yeah. All right. Well, good morning, good afternoon, and good evening, everyone. Thank you for joining us for our September of 2020 Global One Health Initiative webinar, supported by the NIH Fogarty International Center. Our theme for this webinar is the wildlife interface, and we will be hearing from presenters, Dr. Cheryl McCrindle from the University of Pretoria in South Africa, and Dr. Rudovick Kazwala from Sokoine University of Agriculture in Tanzania. And this session will be moderated by our own Dr. Tanya Berger-Wolf, Director of the Ohio State University Translational Data Analytics Institute. And Dr. Berger-Wolf is a computational ecologist, creating computational solutions to address critical questions at the intersection of data science, wildlife biology, and social sciences. And she is also a founding member and project lead for WildBook, which is a project of the nonprofit WildMe, and is an open source software platform that supports the use of AI, computer vision, citizen science, and collaboration to accelerate wildlife research to understand and counter widespread wildlife decline. Dr. Berger-Wolf will provide a more detailed introduction to our speakers at the beginning of the panel discussion. And with that, I will hand it over to you, Dr. McCrindle. You may begin when you're ready.
Right, everyone's ready. Good morning, good. Well, here it is actually just after lunchtime, so good afternoon, everybody. I'm Cheryl McCrindle, and I'm going to talk to you about One Health and antimicrobial resistance at the wildlife interface.
And I would like to introduce you, Dr. Cheryl McCrindle. It is my pleasure to introduce Professor Cheryl McCrindle from University of Pretoria, and she's doing research, who joined the University of Pretoria in 1969 and doing research in diagnostic bacteriology, and then became faculty of veterinary science and went into private practice for a little while in 1976 and returned to academia, 30 years ago when she joined as a veterinary faculty. She received her PhD in 1996. It was made full professor at the University of Pretoria faculty in 2000. Her main field of research since then has been the rural development, zoonotic diseases, epidemiology, and veterinary public health. She also wrote all the modules in epidemiology for animal health technicians at UNISA. And from 2000 until 2012, she developed online courses in primary animal healthcare, participatory epidemiology, elementary necropsy techniques, and milk and meat safety. And in 2004, she was nominated for the Distinguished Women Scientist Award of the National Department of Science and Technology and was awarded the ShopRite Checkers Woman of the Year in Education in 2006. Her publication, The Community Development Approach to Animal Welfare, has influenced the way in which the RSPCA and SPANA and the Donkey Sanctuary approach traction animal and livestock welfare, and she is listed on the FAO website as an expert in livestock welfare. This resulted in many invitations and talks and presentations, and we're very happy to hear from you today, and delighted that you can join this webinar. Welcome, Dr. Cheryl McCrindle.
Thank you very much. I've been to America, and I actually ride quarter horses. That's the other thing I do. So more recently, I've been riding quarter horses in America every now and again. Your lovely country and our lovely country can talk to each other. So let me tell you a little bit more about my lovely country. Livestock in South Africa contribute to food security and rural livelihoods, to macroeconomics, and the GDP. In fact, recently now with the COVID-19 disaster we've had, the only sector that actually did better than normal was the livestock sector, because our general economy went down 52%. And it was actually the livestock sector that kind of rescued it, and the agricultural sector that rescued it, because we were able to produce enough meat and milk and maize and so on to feed our population and to make our, because we weren't importing, our farmers did well and we had a good season. So it does influence our GDP as well. Tropical diseases are a real problem here, and livestock welfare is very closely linked to food security as well as livestock diseases that we have. So you can see this little picture here. There are our different provinces, Northern Cape, Western Cape, Northwest, Free State, Eastern Cape, KwaZulu-Natal, Mpumalanga, Gauteng and Northern Province, and little pictures of the kind of livestock that occur in each of these. Wildlife in South Africa, we use sustainable utilization of indigenous biological resources. The socioeconomic benefits of ecotourism and also managed conservation areas contribute to livelihoods and the economy. So we manage the wildlife in game parks and commercial game farms through culling and good grazing management. For example, as you can see in the picture on the right, the excess males in bachelor herds are reduced with the help of professional hunters, and the meat is harvested for human consumption. I was part of developing primary meat inspection for culled male impala and zebra, because basically we have to hunt these animals out, otherwise there's not enough grazing in the summertime, sorry, in the wintertime in our country. And the male herds take over and push the females and their babies off the grazing. So here's a little picture of me doing primary meat inspection with culled male impala, and we also were in line with the international norms for animal welfare of the EU, so that we could utilize these animals locally and also export the meat. However, antimicrobial resistance has emerged over the last 20 years as the consequence of a large-scale use of antibiotics in both human and in animal medicine. So this results in organisms which we sometimes call superbugs, which are refractory to therapeutic doses, and they can cause major disease and also mortalities in humans and animals. Unfortunately, antimicrobial resistance influences those bacteria which remain in food of animal origin. So if you have meat or milk which is contaminated with organisms that have antimicrobial resistance, this also can be passed on to people who are consuming those products. GOHi is a joint FAO, World Health Organization, expert committee on food additives, and they have principles for the safety evaluation of veterinary drugs, which include antimicrobial drugs. They recommend MRLs or maximum residue levels and determine the methods of analysis for veterinary drugs. They've also established the average daily intake, or ADI, for people who are chronically exposed to veterinary drugs when they are consumers of animal products. So antimicrobial resistance has become rather a question mark for food security and food safety internationally. However, there are challenges with antimicrobial resistance. For example, there are demands for the removal of almost all veterinary antimicrobials, and this can impact on livestock welfare, because livestock in Africa is susceptible to bacterial and protozoal diseases that can only be treated with antimicrobials. Tropical diseases give very high mortality in both our livestock and wildlife. In the wildlife, we just leave them to it, and the lions eat the ones that don't survive. But if you have a small-scale farmer and he is dependent on his goats and they start dying of heart water and he's not allowed to use antimicrobials to save their lives, then his food source is gone, his breeding animals are gone. And so this is quite important. We have very little water in South Africa and huge tracts of savanna, and the grass types are high in lignin, which means that these can be only converted to quality edible protein by ruminants who can digest lignin, turn them into protein of animal origin. And this is a prime source for feeding both the rich and the poor in our country. And it can be applied to both ruminants that are ruminants we have in our backyards, livestock, and also to those ruminants that occur as wildlife. So antimicrobials for livestock can be used for therapeutic reasons and prophylactic reasons, and sometimes also for increasing feed efficiency. Those are mainly ionophores, which aren't used in human medicine, so they shouldn't really have much of an effect on human welfare and health. So over here, I've given a few pictures to show the therapeutic use in South Africa. Heartwort is a serious disease. It's listed by the GOHi as a serious disease of livestock. The vectors are ticks. Amblyomma hebreum is the tick involved, and you can see what a cow in South Africa looks like when you've got an awful lot of ticks under its tail. The agent gets into the bloodstream, and as you can see, it multiplies in the endothelial lining of the blood vessels. And the result of that is the death of livestock, such as Bos taurus, which is the type of animal we normally call cattle, and they die very quickly. We need to use tetracycline to treat the animals, but it's also useful for the vaccine, which is a live vaccine, and we have to actually treat them with tetracycline to block the reaction of the pathogen so that they can become immune. And that's what I've just said here. This became our research problem. Tetracyclines are essential for the treatment and prevention of heartwort in livestock, even for vaccination. They are very cheap and very easy to use, and the withdrawal time is very short. For milk, it's only 5 days, and for meat, it's 28 days. So we had to look, because Jeffco was beginning to talk to us about withdrawing tetracycline off our markets. We allow genuine farmers to have tetracyclines for use against heartwort in their animals. And so we needed to say, well, hang on a moment, we think we'd like to keep them there because maybe the treatment with tetracycline is not the only reason why we have organisms with AMR in our meat and perhaps in our milk. So more recently, there have been a lot of publications about the ecology of antimicrobial resistant genes. But we did this work in 2009, which is 11 years ago. And we knew, at that stage, we knew already that tetracycline-resistant Escherichia coli or Trich bacteria were very prevalent in livestock. We could find them if you just go to the abattoir and you swab the surface of livestock where there's a small splash of feces, we could pick up Trich very easily. The TET genes, which code for tetracycline resistance, we know can be exchanged amongst bacteria, sometimes by bacteriophages, sometimes by unknown reasons. They can go from one bacteria to another, and also between species. And we also know that E. coli can spread tetracycline resistant genes. So the hypothesis was that intestinal bacteria in livestock that were not exposed to tetracycline should not have any antimicrobial resistance genes. So we could look at unexposed impala because they would not have them, and we can compare them perhaps to impala which are getting antimicrobial resistant E. coli out of the environment. And this is how we put this little piece of research together. The aim of the study was to investigate whether Ipicerus melampus, that's the old-fashioned Latin name, in a natural environment which were unexposed to antimicrobial treatment with tetracyclines, but drinking from contaminated rivers, were more likely to contain trich in their feces than unexposed wild impala in a similar environment who are actually getting their water out of springs and natural rainwater. So the Kruger National Park, if you ever come to South Africa, you must come to the Kruger National Park. It's absolutely marvelous. And any of you were there, and give me some sort of WhatsApp or email and come visit. I've got a flat attached to my house for visiting academics. You're welcome to come and stay with me, and we can go together and go and have a look at some wildlife sometime. The Kruger National Park is home to a large variety of wildlife, including impala. We don't treat any of the wildlife at all. We leave them to nature. If they fall down a hole and break their leg, or if they eat a poisonous plant, they just die and they return to ashes, dust to dust, without any human interference. So we don't treat them at all with antibiotics. And we can consider these impala as a naive population. From the map, you can see that several rivers transect the park, and the ones at the bottom of the park run through human settlements. In those human settlements, people use tetracyclines for preventing and controlling malaria. So there tends probably to be an awful lot of tetracycline in human feces that eventually lands in rivers, because many of our sewage plants are not very effective in that part of the world. We have floods that sort of take them out, and sometimes financial problems take them out. So we divided our rivers into polluted and unpolluted rivers, and we looked at data on the chemical composition of the water, which included sampling for bacteria. Then we cultured water samples from the river and found that 3 of the rivers were classified as trich positive and 2 were classified as trich negative. We found that when we collected feces of impala along the borders of these rivers, the impala drinking water from trich negative rivers had significantly fewer tetracycline or resistant E. coli, and those from the positive rivers had significantly higher levels of trich organisms. It was concluded that impala drinking from trich-contaminated rivers were 19.3 times more likely to be resistant. This is a highly significant finding with a P of 0.0001, leading to the opinion that surface waters are an important source of trich. So the AMR in enteric bacteria in both animals and humans could result from trich bacteria that were actually in the environment and not resulting from treatment of the livestock but using tetracyclines. And it also highlighted the importance of a broader definition of water safety for public health, because drinking water could contain AMR genes. This was confirmed more recently by many other researchers, and it also confirms the importance of antimicrobial resistant ecology, which was suggested by the earlier publication we did in 2009. Wastewater treatment can remove or reduce the number of AMR bacteria, but they can't reduce the genes. The genes are not biodegradable, and they can be transferred to other microbes in the environment and in the water. So now we've got to the stage where we even found a publication that says that bacteria that were resistant to antimicrobials could actually be taken up by crops. You've had bacteria from the environment, specifically in America. I do remember cases of salmonella that were dangerous for humans landing up in tomatoes through the water that came out of rivers. And these were actually bad bacteria and they made you sick, but equally using the same theory, if you are eating a tomato that is watered with water containing antimicrobial resistant bugs, you could have antimicrobial resistance transferred to the bugs in your own intestinal system, and that would make you resistant to the use of antimicrobial drugs if you go down with the disease and land up in hospital. So it's very important that environmental sources and the ecology of AMR genes should be further investigated. And we really need to start using risk and hazard analysis for antimicrobials when we talk about a One Health approach. It's absolutely important, I think, not to use antimicrobials as growth promoters. And we should also really consider livestock welfare before we go taking particular antimicrobial drugs off the list of those that can be used by veterinarians and farmers. So I plead the case there for tetracycline, because tetracycline is not really used very much in human medicine except for the control of malaria. And it's a very cheap and easily obtainable drug and very useful. We also use it for treating salmonella in backyard poultry, and I was recently asked about that by someone from another African country where they have a tremendous amount of salmonella-linked syndromes in children that are eating eggs and eating poultry meat. So basically tetracycline is a lot less dangerous than the salmonella. So I've got here all the key references. I'm not going to go through them. Newer ones are Graham et al. And Kochman et al. And we also looked at whether the transfer of antimicrobial resistance could be a new form of zoonoses. So I'll just whistle past those. I thought it might be interesting for you to think about a few other things which are other One Health aspects of African wildlife. The picture on the right is a foal belonging to one of my clients. It was taken by a leopard. We actually have leopards on our own farm. So you can see the typical way in which a leopard predates on livestock. They leave the legs and the head, and they just eat the intestines and the abdomen. So we later got the same leopard captured near to us because he was being a bit of a nuisance and eating people's dogs, and he's been rehomed to a proper game reserve where he can go and predate on things he's supposed to predate on. So jackals and the other wildcats such as caracal and cheetah and lions are also a bit of a problem when they start predating domestic animals. We have crop damage by elephants and buffalo, warthogs, wild pigs, and even wild birds. We have human-animal conflicts at the wildlife-livestock interface, particularly elephants that tend to tread on people and lions that land up in people's villages. Then we've got zoonoses like Rift Valley fever, rabies, and of course we've got West Nile fever, which has just appeared in our country and apparently affected some flamingos. It comes in seasonally. Notifiable diseases have quite an impact on our economics. Foot and mouth disease is endemic in buffalo, and it spreads over to livestock, and then we can't export any of our beef or pork or mutton. African swine fever is endemic in warthogs and in wild pigs, and you know about the huge epidemic of African swine fever that's just happened in China. Then we've got avian influenza that periodically comes in with wild birds, and that's another problem with absolutely stopping all our exports of chicken until we've sorted that out. Then, of course, we have droughts and fires and floods, which also affect human and animals, like the terrible, terrible fires you have at the moment in Iowa, I believe, and parts of America, which are so sad and so huge you can see them on satellites. So that's an international problem. We also had the same problem in Australia. Right, well, thank you. That's what I have to say. If there are any questions, I'm happy to discuss them.
Oh, thank you so much, Dr. McCrindle, for your wonderful presentation. We will actually hold questioning for the panel discussion. after Dr. Kazwala presents, or yes, so thank you so much. And Dr. Kazwala, if you're able to share your screen, that would be great.
And while Dr. Kazwala is is sharing the screen and setting up, I will introduce, and Dr. McCrindle, I think you need to unshare, stop sharing.
Unshare something.
Stop
Stop
sharing.
share. Oh, you are so clever. Thank you so much. Okay.
Yes, and so now Dr. Kazwala can share his screen, and I will introduce, so Professor Rudovick Kazwala is a graduate of Sokoine University of Agriculture. He MVM from University College Dublin in Ireland and PhD from University of Edinburgh in UK. He's the Professor of Ecosystems and Public Health in the Department of Veterinary Medicine and Public Health, College of Veterinary and Medical Sciences at Sokoine University of Agriculture in Morogoro, Tanzania. Professor Kazwala has supervised to date over 45 postgraduate students, published total of over 100 articles that are available through PubMed. Most of them are available through PubMed online. He's the chairman of the Veterinary Council of Tanzania, a member of the International Health Regulations roster of experts in veterinary issues, zoonoses, and coordinates research on bacterial zoonoses, viral zoonoses, and foodborne pathogens in various networks in Africa. For the past 1.5 decades, Professor Kazwala is the Tanzanian Director of Health for Animals and Livelihood Improvement Project, a collaborative research between College of Veterinary Medicine at University of California, Davis, and Sokoine University of Agriculture. Since 2009, Professor Kazwala has led the USAID Emerging Pandemic Threats Program, PREDICT project in Tanzania, supporting the development of wildlife disease surveillance and early warning system for high-risk viral pathogens, and heads a new molecular laboratory for emerging infectious diseases and pathogen discovery. From 2014 to 2018, this work was carried out in tandem with the DTRA-funded projects, such as zoonotic selected agents in Tanzania, One Health approach to Rift Valley fever and Brucellosis surveillance, and evaluating zoonotic viral sharing in South Tanzania. Professor Kazwala has recently been recognized and awarded by the Tanzania Wildlife Research Institute and Tanzania National Parks for his excellence in research in wildlife disease in Tanzania. His own university, Sokoine University, has also awarded him the Edward Moring Sokoine Researcher of the Year for Excellence in Research in 2019. And with that, welcome Dr. Kazwala, and we're happy that you can join this webinar and share your research. Thank you.
Thank you for the introduction. Good morning to all, and good afternoon to those in Africa. As I've been introduced, I'm at the College of Veterinary Medicine and Biomedical Sciences. I hope you can hear me very well.
Yes, indeed, loud and clear.
Thank you. So my topic is based on what you requested. I'll be working mainly on the wildlife diseases which you have encountered in human livestock wildlife interfaces in Tanzania for a number of years. So we need to just remind ourselves why disease occurs. And I think most of the participants are aware how the disease occurs. The pathogens are either intrinsic or they are introduced into the ecosystems, and therefore they will affect the appropriate hosts. And in that interactions, we may have disease conditions which are either fatal or mild, or we may call subclinical, but also those infected may be carrier of those pathogens to other species. So why diseases in wildlife? I think the issue is that these interactions which occur between the host and pathogens may be influenced by various factors, as you can see on my slide. In nature, if we let nature take its course, we have situations in various places. I've copied this from Bill Kadesh's paper. You will see that from the bottom left that in the green zone is the wildlife, and disease may be occurring in low instances and changing patterns at some point. But when this disease in wildlife, they get into livestock or domestic animal populations, there might be some amplification aspects, and they may infect a number of domestic species and cause morbidities and mortalities. But at the same time, these pathogens from wildlife may get into human populations, and the same amplification may occur and we get mortalities. But when the disease is amplified in the domestic population, especially for zoonotic diseases, they may get into human populations and you get mortalities and morbidities. This is just like a natural cause if we don't make any interference. So where are these emerging infectious diseases coming from? We see that in the past decades, and here is a little bit old data, but you see that the proportion of zoonotic diseases affecting human coming from wildlife population has increased. You can see on between 1985 to 1995, the proportion was much higher. And which animal populations are of concern? Some studies have been done, and it has been shown that there are certain taxa of animals which are more responsible for the diseases in human, the zoonotic diseases. You can see here the bats, rodents, and non-human primates are being considered to be higher source of the pathogens to humans. And as have been introduced in one of our work, the PREDICT work, we did a lot of sampling of non-human primates, rodents, and bats to determine if there are viral pathogens which can be shared between humans, between animals and humans. We can classify wildlife disease into 4 categories. Some of them are purely pathogens in wild, and they go and infect humans. Some are pathogens affecting the domestic animals from wildlife. Some of them might have just importance on the wildlife populations, but some of the wild diseases have effect on the ecosystem and the functions of the animals in the ecosystems. There are a number of diseases which we have encountered in our countries, from wildlife to domestic and animals and humans. I'll be discussing some of those which are mentioned here in various ways. How do we know there are wildlife diseases? It's through disease surveillance. And on the brief on my bio, I mentioned some of our 9 or 10 years work with PREDICT, which was on disease surveillance on those non-human primates. But before that, we worked on large ungulates and buffalo. And we looked at a number of diseases at different times of the year or different decades. And some of diseases which are mentioned by my colleague, Dr. McCrindle, rinderpest has been one of the diseases which we looked at. Rift Valley fever, foot and mouth disease, tuberculosis, bovine tuberculosis, and brucellosis. It's through disease surveillance where we collected samples systematically and they do the analysis for a range of a period of time. Or could be on purely research. Like here, these are 2 of my PhD students who were working on buffalo. One was looking at tuberculosis in buffalo, and the other one was looking at brucellosis in buffalo. So through that, we can gather information about the presence of certain diseases in wildlife. But we do conduct postmortems, especially where there are kills of wild animals for intention or non-intentional. We can look at a number of pathogens in those animals, and you can realize which diseases are present in those animal species. For example, in our work in various ecosystems within the country, sorry, I didn't provide the map, but this is like the west of the country is the Katavi, where we looked at livestock and wildlife. It's mainly livestock and buffalo. Then we had the Mikumi-Selune ecosystem, which is almost in the center of the country. We looked at livestock and buffalo. And then in the Serengeti ecosystem, also we looked at tuberculosis. And we found that in the various levels of prevalence. But we had done some work on other pathogens, and we are prompted by mortalities in rhino in the year 2001. It's a long time ago, but we linked that with some vegetational changes, climate change, and they will have to do some studies on ticks to see their involvement in the mortalities. In warthogs, especially in search of African swine fever, we capture them, we take the ticks, the soft ticks, and we try to see if there are problems of swine fever, but also trypanosomiasis. Among the diseases we looked at in our works, we looked at the transboundary disease, these diseases which are reportable as required by GOHi. And one of them is malignant catarrh fever, where we know that wildebeest are reservoirs, but this disease may infect cattle, especially during the lambing season of the wildebeest. And in our ecosystem, especially in around Serengeti and Ngorongoro, you find that there's mingling of domestic animals, as you can see in those images, with the wild animals. And that can lead to transmission of diseases. As already mentioned about our work in Budapest regarding wildlife, ruminants, and cattle. And already I mentioned about swine fever. And all other diseases, including foot and mouth disease, which is the most recent work. We found that while we consider domestic cattle to be the major animal affected, but we have reservoir or animals in the wild also affected. And we have one of our publications in the ecology, nature ecology, where we found there are waves of foot and mouth disease within Eastern Africa. And that could be a way we can look at vaccination strategies for foot and mouth disease. Sorry. The other is when we had avian influenza globally, also we had to do some work in our part. Luckily, avian influenza did not transcend to our country at that time, but we are looking in wild birds and also in poultry, trapping some of the migratory birds and doing sampling and doing analysis in our labs. Then we have diseases of public health importance. There are zoonotic diseases, which also been mentioned by my colleague earlier on. Rabies, anthrax, sleeping sickness, all the way to some of the zoonotic parasites, especially trypanosomiasis. Rabies is of major concern, and we had some issues, major issues on rabies in our part, where at some point we thought it will lead to depopulation of the wild dogs. And some studies were done in and around Serengeti National Park with colleagues from University of Glasgow. Professor Sarah Cleveland led this work, and they could see that there were some kind of a pattern or a trend of the disease when rabies was introduced in around Serengeti. The wild dog population declined. And this was the trend around that time where we could see a decline in the wild dog populations. So that triggered us to do some work in terms of vaccination in and around the park to prevent this rabies spread. The scenario was in the western part of the Serengeti, there was high domestic dog density, and the eastern part, there was low. And the trend could show that where we had low, still wild dogs could be observed, but where we had a high domestic dog, there was a few number of these dogs in that national park. And the vaccination was introduced, and the trend was reversed in terms of population of the dogs. Brucellosis has been a disease of concern for many years, especially on the economic losses in terms of abortions and calf mortalities. And we have looked at that in the ecosystems. And we looked at cattle and looked at buffalo, and we could see some signs of disease in some of the wild animals. Like these buffaloes in different ecosystems, they had hygroma, which is very suggestive of Brucella. And actually, for one on the top right, we captured this buffalo, and we extracted the fluid, and it was full of Brucella abortus material. So we can see from through those research, we could show how the disease was spreading in various national parks. This was mainly in the Serengeti National Park and the Ngorongoro Crater Authority. In the Rudovick ecosystem, which is another national park, which is in the southern part of the country, also we found the TB and the brucellosis among wild animals. Anthrax has been a problem for many, many years. And if you look at our history, here are the national parks and game reserves where it has been reported. But you could see at the bottom there, the Serengeti ecosystem, where anthrax has been reported year in, year out. So it's a big challenge in some of the ecosystems. And a number of, from cattle to wild herbivores, they are all affected, including elephant, giraffe, buffalo, zebra. Those all animals can be affected, and we had a lot of that. Human in those populations. In this Ngorongoro crater area, we have the cohabitation between wild animals and domestic animals, where domestic animals introduced into the crater, the grazing land, and there's salt water where they can graze for a short period of time, but people were keeping those animals living around there. And when we had the anthrax case, this is the situation we saw. Of course, there are some issues regarding anthrax in those populations because of the behavior and the culture, where people are not willing to destroy the dead animals. They would like to have some meat from them. And therefore, authorities have to intervene and burn carcasses and also treat the cases in human. Sleeping sickness, human sleeping sickness has occurred, especially in the western part of the country, but there were some few cases in Serengeti where especially tourists who are visiting there, they got a disease, but this has been contained. We don't see any more sleeping sickness in around Serengeti. And of course, we don't see, as I said, what's happening now. We don't see that disease. Rift fever was also of attention in around 2014. We did some studies, and they looked at the inter-epidemic period, and we could find some traces of the disease within the pastoralists and in some of the wild animals. Exposure in terms of IgG could be very high, especially in the antelopes and the buffaloes. Some other parasites which are of importance for wild carnivores were also part of our work, where we found them in various ecosystems. Other diseases of concern is the canine distemper virus. It also had its share on the deaths of lions in the Serengeti, but also with rabies and canine distemper for the wild dogs. This was a big problem. We have a problem of, this is on canine distemper, and the outbreak which I reported in lions, and we had an epidemic around that time. Then we have the giraffe disease, a skin disease, which is, you can see lesions on the extremities. And we did a lot of work to see what's involved. And we found some worms involved in these lesions. Right, you'll see I'm part of that team investigating this problem. Treponema has been also a problem which we investigated in some of the ecosystems. These are baboons in the national park, which are having a treponema disease. And we looked at what is the actual pathogen, which treponema species is involved. Of course, the diseases are in the genital area, could be thought of syphilis, but syphilis is not the case. We found treponema peteneus as a pathogen which are affecting these non-human primates. And the lesions could be similar to yours, as you can see on the bottom right for the non-human primate, but on genital lesions. The top pictures are normal and the bottom pictures are abnormal. And we found this disease to be spread in a number of the ecosystems within the country. Lesser flamingo was also a problem in one of our alkaline lake. They died. We don't know what was really a problem, but we thought algae could have been involved. Zebra, we had the issue of equine herpes virus, and this was investigated thoroughly. Of course, there were also a lot of worms in these buffaloes, so we thought probably it is a combination, not only of a herpes virus. But in our work, not only to investigate the presence of the known pathogens, but we need to also to look at unknown pathogens. Based on my earlier slide, where we are showing to share highly with these taxa. And we did a lot of work in wildlife surveillance for non-human primates. We did non-invasive studies using fecal material, and they looked for a number of viruses which could be found in the non-human primates. We did a lot of sampling of bats in thousands, and we capture them, and then we take samples for analysis and look for viral pathogens which are of concern. And we use as I've been mentioning, I have a viral discovery lab where we are looking a lot on those pathogens. And the major viral families we looked to are corona, not COVID, phyllo, flavi, influenza, and paramyxo. These were the main viral pathogens we looked at. And through these studies, capturing bats and doing some analyses, we found, especially corona alpha and beta, we found them in fruit bats and the small bats. and also paramyxoviruses were also detected in some of these fruit bats. Some of the findings on emerging viruses, these are, I could say, the new viruses in terms of species, because when we isolate the virus, we do some analysis and we get sequencing and blast on the database. You could see they are not linking to any. So we named them, and these are some which have been approved already. But in order to do a lot of studies on possibility of transmissibility and the spread of the viruses, we looked at the fruit bats. And we did a lot of work in terms of capturing and tagging them, putting them in a satellite transmitter, and you could monitor their movement where they are roosting and where they are feeding themselves. These are fruit bats. They travel large distances. And we could see through satellite data where are these animals going to. And here is one of the examples. They go into some places where they are urban, and they go into the forest, mainly in search of food. And now, if these bats could be harboring some pathogens, it's likely they can spread easily throughout the country and in other places. We did look at where these bats are foraging, or they are finding the fruits, which other animal species or which other mammals goes into there. And you could see through these cameras, the infrared, you could see a jackal, but also in the morning, you could see that non-human primates, they come and they take the remains from the bats. So it's possible for viral transmission. Yes, just the network of and the population densities. And if, for example, we have in one area a viral pathogens, then it's easier to spread through the road networks and populations which are in the country. And this was a model we tried to show the spread from our place where we're doing research. If a coronavirus spread, how fast it could go into different angles of the country. But we have other, not only disease, but other conservation challenges, which are due to the interactions. For example, the littering of areas where we interact, the animal feeding, roadkill, disturbances, and fires, and the introduction of exotic, especially plants, into the national parks. And here are examples of animals which have been killed on the road. This is a pangolin was killed on the roadside. Here we have an eland which was also killed on the roadside. Here is an impala which was killed on the roadside. The elephant was killed on the roadside. And the other, we may introduce other pathogens through our handling of animals, whether intentional or accidental. Here is the activities of capturing wild animals, and these may, in one way or another, introduce the pathogens to the wild populations. So what we need to do is to address these problems in a One Health approach, and where we could deal with the problems at an early stage, early warning systems, early detection, and the forecasting. If we forecast and do detection early in the wild population, then we won't have any spread or amplification in the domestic animal population. And you can see the deep blue colored area is the reduction because of the intervention. And if we do intervention in the livestock population, even in human, it will be decreasing. So in the end, we'll benefit from a One Health approach. So that brings to the end of my presentation. And we wish to acknowledge my colleagues whom we have worked together for a number of years on these issues of wildlife disease in Tanzania. Thank you for listening.
Thank you, Dr. Kazwala. And thank you, Dr. McCrindle. So we now move on to our question and discussion portion, and please type your questions in the Q&A box, and we will I will read them out and we will answer, and then feel free to add questions as the discussion progresses. So we already have a couple of questions, which I think summarize both of your and can be addressed to both of you. So I will start with one general question and then move on to the ones that are already in the chat. So you're both addressing this interconnectedness of global health, global One Health. You know, the connection between environment, wildlife, domestic animals, humans, and you know, this intersection with global supply chains and travel. And you're both looking at different perspectives. What of your approach generalizes beyond your country, your system? What can we learn from each other? And I will start with Dr. McCrindle.
Well, actually, I think I've learned a tremendous amount throughout the world, because I have been working with the Food and Agriculture Organization previously, and I went all around the world with the International Dairy Federation. And I do see there's a huge amount, for me, that almost is what brought One Health in. I'm now with the School of Health Sciences, and I think our COVID-19 experience is a very, very, how can I say, expected thing. We knew this had to happen when the first SARS came along. And I'm very interested in what Rudovick Kazwala is doing as well, because this whole interaction, we have to save the planet, and the planet is an ecosystem. And we can't just say, well, people are more important than animals and trees and plants, because if people are all by themselves without trees and plants and animals, we probably wouldn't survive. So we have to look after each other, and we have to watch. That's what's so attractive for me with the One Health system, is what happens in your country. I sit and look at, recently I saw quite a lot of people shooting grizzly bears because they're going out and getting in the way of human activities and threatening human life, and gave you the example from my country where we had a leopard who was fairly dangerous. He could have taken a child. He took a foal. We found him, we shot him, and we put him, well, we didn't shoot him with a gun. We shot him with something that could preserve his life, and we went and dropped him in the local game reserve, which has got enough prey to keep him busy and for people to look at him. And again, we look also at the way we're managing, which I mentioned in my presentation, that we're managing our wildlife. We have beautiful wildlife preserves, but we don't leave it to nature now. We manage it very, very scientifically, and we have highly sophisticated science into exactly how many of the bachelor herds must you cull. And before you start culling, you need to look at which of the rams you don't want to cull, because some of those really good rams live as long as 30 years. So if you know your animals, you're not going to kill that good ram who's looking after his herd so well, and you rather go and cull the younger animals, which you also can market more easily because they have a better flesh. And then you can also look at interbreeding within livestock herds. We're having this in some of the game reserves where the livestock is the wildlife is so interbred that you've got to shoot out all the bulls and bring other bulls in. And these are the things I think that where the wildlife interface, antimicrobial resistance is one of the minor things, but this whole One Health approach, an ecological approach, is something I'm passionate about.
Thank you. And Dr. Kazwala, your thoughts on what can we learn from each other, and what you clearly already are learning, and what can be learned from your work?
Yeah, I think we should know that the pathogens exist in the ecosystems, and no one part of the ecosystem can maintain those pathogens. They depend on various factors within the ecosystem. And therefore, when we are addressing the problem of the diseases, we have to address the issues of ecosystem. where are these pathogens surviving? Where are they? I mean, when you look at our work on bats, we could find coronaviruses, of course not COVID, but coronaviruses in a number of situations. So these are living, the corona are living happily with the bats. The moment they jump from one taxa to another, that's where the problems occur. And I think this is quite evident of what has happened, the COVID situation, that we think it's bats and then move to pangolin and the others, and then to move to human, it found a suitable host for expressing its virulence, and therefore we have a disease. So we should be knowledgeable of what's happening in the ecosystems. And as Dr. McCrindle said, there's no one medicine for addressing these problems. We need to work together. The One Health approach is really something which we need to embrace in various ways. If you look at the situation of bats with those pathogens we mentioned, you need to think about conservation. Bats are important for various functions, control of various insects, pollination, and so forth. But at the same time, they may be bringing another challenge to the human population. So when we work in our situation, we need also to work even to social scientists who are able to know why people are preferring certain habits and how they view the wildlife and how they interact. This is very important. So a solution to these pathogens occurring in various situations is really a One Health approach.
Thank you. Moving into the questions from the audience, Dr. McCrindle, question from Nega Asamene, who wants to know your country, South Africa's experience on AMR surveillance using One Health approach in South Africa, and whether there is common database for AMR for human, animal, and wildlife, both domestic and wildlife, that is available. I want to expand it to a broader data question. So of the sort of this big system around One Health, can you comment on the availability of data and the data gap, perhaps?
Well, one of the wonderful things about being an academic is there's two kinds of data. There's data somebody else collects that you can make sense of, and there is data that you've got to go out and collect yourself. The data we collected on the Impala was great fun because my little colleague came from, my student came from Italy, and she went in and she had a game guard with her because it was so dangerous, and there she was right in the Kruger Park picking up feces of animals, and we had it all scientifically planned, and it was great fun, and that was new data. But we also have quite a good database on livestock and wildlife populations in South Africa, which you can access in different ways. So as you have elsewhere in the world, and if you even look in Kenya, I think Rudovick has some very, very good data there. He had fantastic stuff on bats, as I saw. I don't know, Rudovick, what you feel about that. No, I mean, how you feel about this question of, new data that we as scientists have to find, but sometimes there is existing data on wildlife and humans that you can actually tap into and do new research with old data.
Yeah, I think it's access to the data is a challenge, and also storage in a way that it can be accessed. There has been a lot of work, especially, for example, in wildlife. If you go to the parks, they've got some records of many, many years, but how do you access them to give you information which you can find a gap to address by research? That has been a challenge. The data is in gray papers, and maybe in a books, files, reports, and you are not able to access them. So access to the data is a challenge. You may be doing a research on something which already someone has reported, but it's not coming into the public domain. But also, there are challenges of legal aspects. Some data may not be allowable to be shared, especially when you have diseases of major importance. Country may want to keep them, especially when you talk of wildlife sector. They don't want to scare people who are coming to visit, tourists or whatever. So some data may be captured and stored and not released. So we have some challenges on how to access data in that situation.
I think it is a big challenge in general, data access and data sharing, that is now being addressed in many contexts, both scientific and health, across the world. For United States, there is a requirement for availability of research data, and there is a federal mandate that requires access to all research data sponsored by all federal agencies, as well as from all major publications. Access doesn't mean disclosure for anybody, but protocol for having access, a clear protocol with every publication for having access to data that supports that publication, which is starting to improve things a little bit, I think. But as a data scientist, I very much agree with the statement that data sharing and common platforms and procedures and protocols for sharing data, particularly when we have different jurisdictions and policies around data sharing and data availability and reporting, access, privacy and security. And we're starting to talk also about the wildlife endangered species, privacy and security. So we don't accidentally disclose information that wipes out the entire species in the process. That is also important. I do want to circle back briefly to the specific question on AMR surveillance in South Africa and whether that data is available, Dr. McCrindle.
I'm pretty sure we have some data on it. Where exactly, I don't know, but we do have a tremendous amount of data on antimicrobial resistance, and it is shared internationally because we are members of GEFCA and they have access to it. So we probably, I have seen in various publications, you know, some systematic work which says surveys of different antimicrobials in South Africa. I think I vaguely remember seeing one for this year somewhere. Of which ones are where, and GEFCA I think would have some of that data. If somebody particularly wants it, I can go. What do we do? It's a bit like going and finding a wreck with treasure in it, you know. You sometimes have to sort of go and have a look for data. But if somebody's really interested, I'm sure I can put them in contact with people who might be able to assist.
Thank you. And I would like to introduce Dr. Rudovick, yes, Director of the Ohio State University Global One Health Initiative.
Thank you so much, Tanya. But really great, lots of wonderful discussions for your moderation, and great to see Cheryl and Rudovick. That's great. Lots of very important points entertained here. So very much appreciate presentations and the discussion. I have a kind of quick question. Definitely realize South Africa, the Republic of South Africa as a country, is one of the really leading, I think, in sub-Saharan Africa on antimicrobial resistance, data generation, and other aspects. So for you, my question is, like I know from UCTs or Mark Mendelson and your Department of Natural Resources, they call it different data. I have different colleagues from the human, environment, animal, but how much that interface, the One Health connectivity, maybe if you can address that. And my second maybe broad question for both of you, and in Tanya included, many of you working on wildlife interface, as the population of the world, but particularly sub-Saharan Africa grows, in that interface, wildlife and domestic and human is getting closer and closer, do you see the resources available to collect data, to generate outcomes, to make impact? Is it in pace, or what do you see in terms of generally the resource needs and how the world is? Maybe I'm sure you will be better informed to tell us. So thank you, those two questions. Appreciate it.
With regard to the first question, I'm with the University of Pretoria, and I was a lecturer in veterinary public health, and I just happened to be very good at zoonoses, and I managed to find all sorts of pictures of zoonoses, and I got kidnapped when I retired by the School of Health Sciences and Public Health, and I lecture there on zoonoses. And I work quite often with other experts. We have some very good experts there, particularly on, we've done some tremendous work on West Nile fever at the moment. I was listening to a seminar last night. We've got a lady in our department who's a virologist. She's under the Faculty of Health Sciences, and she's Mariki Fenter, and she's done some wonderful work, really, really fantastic work on the West Nile fever and related viruses and the hemorrhagic viruses. So we've got, she's actually one of the people managing One Health. That's one of her focuses in her research. It's one of the focuses in my research as well. I'm with the National Research Foundation as a rated scientist. The University of Cape Town has got people as well. I'm aware of that. VITS, we're working with NICD, the National Institute of Communicable Diseases, and the VITS people work quite well with the University of Pretoria. We have joint research things. I think South Africa, I don't know what to say about funding. I don't get funding. I've been forbidden to go looking for funding because I'm over 60, and nobody over 60 is allowed to look for funding. So I get very creative. I run a veterinary practice, and that kind of subsidizes what I do. And then occasionally I ride horses for fun. So I'm with the American Quarter Horse Association, and I pop over and ride over at the American Quarter Horse Association and talk to some American colleagues at the same time. So you have to be incredibly creative to get funding in South Africa. It's not easy. We do get Bill Gates, gives us quite a lot of money. World Health Organization helps us. We're members of OIE. You can go and look at FAO. I get money sometimes from ILRI, International Livestock Research, and that's where Rudovick's also, he's very much more important than I am there. And they very kindly took us up to the African Union. And honestly, I would say Americans should not be afraid to come here. We find a lot of Americans saying, I'm dead scared to come to South Africa. Well, darn it, I have been right into the Bronx, and I've been into some of the ghettos in Washington. And your country also has its problems. If you smile and you don't wear too much expensive jewelry, you're fine in Africa, just as you're fine in the Bronx in New York. So I'd say we're both of us are part of the same Southern African collaborative thing. We SADC, we're both members of SADC. I don't know if that helps at all. Have I answered your question on that?
Thank you. Yes, Cheryl. Yeah.
And then I think the second question, I think, could Rudovick answer that first, and then I can maybe add to it a bit?
Yeah.
Yes, thank you, Nusen, and it's nice to see you after a long time. The issue of funding to do some work in the interface depends on what actually is the issue. I gave you the category of those diseases in the wildlife where it depends. If you are addressing a problem which is touching human health, you will be getting funding quickly. If it's addressing a transboundary disease, and it's going to affect other countries, you can get funding. But if it's a disease which is endemic, nobody will provide resources for that, okay? You will struggle. So we have seen, for example, but maybe I should say, with One Health now coming in and becoming an approach, and we have platforms within the country for One Health, definitely things are changing. We are realizing, though these are neglected diseases, but the impact on the livelihood of people in those areas is tremendous. The people living with the Brucellosis, animals aborting, livelihood is impaired, then things are changing. But from the international perspective, it's mainly when you have disease of high impact, especially to human health, you will get some resources for doing that.
I think there might have been a second part to your question that we haven't answered. Could you maybe just repeat it to?
Yeah, related to that, but yes, thank you both of you and Rudovick in terms of, yes, it really varies on the issues, but with the overall trend of this interface being more and more intensified. And, I give credit to World Bank colleagues, Frank, Bert, and others. Just, 2018 or so, there was a publication, for example, highlighting the economics of One Health and this interface, and the world should invest about $4.3 or so billion a year. Mm-hmm. And that was, of course, we know it, that not that much is invested directly for One Health purpose. But my question is, considering this intensification of the interface, is the resource allocation overall? That's what, Rudovick touched it very well in one way, in definitely an important angle. But do you see the trend? We know each other, right? There is in at least the past 10, 12, 15 years, we see more and more increasing outbreaks related to One Health, related to wildlife, emerging, re-emerging infectious diseases. Do you see the resources in pace with that, the overall allocation? Yeah.
I'd like to just add there, I think, one of the things that has just been mentioned by Rudovick was brucellosis. Brucellosis for me is a very scary disease. I do a lot of work on brucellosis. I'm busy publishing on it because a lot of our data on brucellosis was embargoed. And I mean, one of the frightening things I can perhaps mention to you is we did recent work with the wives of men working in abattoirs, because I've seen, I'm telling you this because I'm just telling you, I've seen some of my female veterinary colleagues having trouble having babies. You get lots and lots of, you abort your babies if you have brucellosis. You get miscarriages one after another, and it's never diagnosed. So we went out and we were talking to the wives of men who worked in abattoirs, and we discovered the literature says it can be genitally transmitted, but the percentage of their wives that are having repeated miscarriages is excessively high. Now, brucellosis is a worldwide phenomenon. It occurs in wildlife and it occurs in livestock. And this is a research thing which you just cannot find funding for. We went and had a look at it, and people all sort of shrugged their shoulders and said, well, it's not true. Actually, the literature doesn't say it's really important for that. But as a woman and a mother and a grandmother, I feel the fact that women are losing babies because of brucellosis and not being treated in time is very sad. And also, it also, as a veterinarian, it causes quite a problem with back problems. And I have a number of colleagues whose spines have given in with brucellosis. And over the time, it's also been kind of shoved under the carpet. So you have to be very, I'm probably getting on my high horse again, but probably I think one has to be very careful, because there are fashions in things that you want to look at. At the moment, everybody wants to do coronavirus, everybody. But there are a lot more diseases than just one. That's my bit.
Yeah, no, great point, definitely. Already, even to qualify, Rudovick, I was thinking that you mentioned, even within those zoonotic or human health importance, there is discrimination among the diseases in terms of resources. I think we have to be honest. Yes, of course, now the fashion is COVID-19, but even beyond COVID or before or after, as we know, brucellosis is highly neglected tropical disease, not much interesting for pharmaceuticals to do anything. I had my veterinary, one of my professors in Ethiopia, had brucellosis and he doesn't have children. In fact, infertility, and men also impacted, including, of course, women. But when it is highly contagious viral diseases that could go across the planet, that could impact the developed world, clearly, many governments, everybody focuses on them, and it impacts the various demographics of human health broadly. Yeah, then you're absolutely, I agree with you. That's a very good context to see. So thank you so much. I don't want to take too much time.
I want to add very briefly sort of a very different dimension of understanding the getting the data on disease surveillance and the human-wildlife interface from different perspectives. And that's, so my view into it is really creating artificial intelligence and remote sensing tools for getting data. And Dr. McCrindle, you mentioned you've been looking at pictures of diseases. So one way to really get, maybe not perfect, but at least the baseline quick data is through images, right? So the system that we built in general is for wildlife monitoring is through images, and the images from any source of, whether it's scientists and field assistants, camera traps, drones, or even tourists going on safaris to all the wonderful African continent wild parks, and getting that information quickly. But there is a lot more that's now being built, and to get the basic data on how many animals, where they are, what are their conditions. So we often get requests for animals to see the scarring, the different decisions, deformities, health, so you can quickly at least assess at the population level. This is hopefully just in aid for the wonderful work that Global One Health is doing. So Dr. Kazwala, you mentioned rabies in wild dogs. Wild dogs, as you know, are individually identifiable easily by the pattern. And so we're building automated systems. And in fact, Wildbook for wild dogs is going up live, combining data from ranging from Botswana in the south to Kenya in the north, and the continuum of the range of these animals to understand the population dynamics, and then it can be linked to diseases. So I think there are some aspect at least of surveillance can be done through technology, clearly not all of it, and we have to work in partnership. With that, technology and data are only as good as the actions that we can take based on that. And so I want to move on to the question that is from the audience, from Ngusu Fasil. What measures are critical in terms of BTB and MTB monitoring and evaluation of wildlife livestock human interface? And to broaden it a little bit more than that, so how do we understand from data, from work that you both are doing, what is actionable, what measures are critical, and how do we assess the actions of those measures? So maybe we start with Dr. Kazwala on this, and then Dr. McCrindle. You're muted, Dr. Kazwala, so you need to unmute. Yeah.
So I'm saying I had interruptions. There was a mobile phone, so I could not get the last bit. Sorry about that.
So no problem. So the question is, what measures, the specific question, what measures are critical for BTB and MTB monitoring and the evaluation in wildlife livestock human interface? And more broader, how do we move from the data and evidence and research to actionable measures, and how do we evaluate the effect of measures that we're taking at this wildlife livestock human interface in terms of disease mitigation?
Yeah, I think as also it was mentioned earlier by my colleague, the issue of wildlife, intervention in wildlife is still an aspect which is you have to go through a lot of regulatory aspects in order to make intervention in wildlife per se. Using the example of rabies, which I mentioned, there was this issue in Serengeti. We went around by doing control in the domestic animals. So if you put measures in domestic animals, therefore you reduce the instance within the wildlife population. For the bovine TB, well, in our country, we just wanted to show the evidence, the presence of the disease in the wild, but there was not much in terms of control of bovine TB. Even in domestic cattle, we don't have national programs to control the bovine TB. So sometimes you can do a very nice work to show there's a problem in wildlife and the problem is going into the domestic animals. It depends on the nature of the disease. You may not go further in terms of intervention. But what we were doing was to make the communities aware of the challenges which are occurring in that situation. And they can make their own judgment in terms of, or decisions in terms of what kind of intervention. Of course, for bovine TB, we talk about milk to be a major source to people because they don't boil milk. So you have to educate people to boil milk. Because if you tell them to cull an animal, that will not sail. So you need to put them on measures on food safety so that they can control the disease. They wouldn't match. We asked a lot about if they are knowledgeable of zoonotic diseases coming from wildlife. 100% they don't have. But when you talk about probably some diseases like anthrax, from domestic animal to people, they will tell you, yes, we see this and that and that. But for some of these, let's say, benign diseases, chronic diseases, they don't realize the effect is coming from the animals they live with. So again, it's dealing with the populations which are surrounding those places, providing local solutions to a local problem. That is what we are mainly advocating. I remember one of the population, one area nearby Serengeti, we were doing the TB testing. But before we started, we informed them, we are going to be doing a TB testing. There was a meeting of the villagers. We explained to them what is really the problem and what is the consequence when we find an animal reacting to TB. And it was taken up by the village council and they agreed. Whichever animal is found to be positive in this community has to go for slaughter. So it was a community-based approach, which made it people to accept. But sometimes you need to go on the medical grounds. You see, this is a problem, people are suffering that, and you need to do A, B, C, D. So that kind of intervention we can do.
I think you're absolutely right that community buy-in is an important aspect. And so, Dr. McCrindle, if you have a few, we're coming up on the hour, so if you have a quick insight to add to this, and then we'll move to wrap up.
Well, yes, basically, I think the important thing is that MDR TB is actually bovine TB. It's multiple drug resistant. We've got XDR as well, but the multiple drug resistant TBs in our country are often as a result of bovine TB infection. There was some very good work in Tanzania where they found that human TB was being transmitted. Theoretically, it can go to a cow, but it's not transmitted back to humans. But we found that in those publications I read, it was going because the people milking the cow were coughing at the cow became infected in the lungs rather than in the udder, and then they were coughing back at people. And that was also quite interesting because we found that human TB also plays a role. If you've got a very high level of herders with human TB, you get false positives on your bovine herd for BTB, and then you've got to slaughter animals which don't have BTB. It's very good. The XDR TB is a big problem in us with the mines, so the miners have a fair amount of XDR TB in our country as well, and TB is a huge problem in our country, but bovine TB is a listed disease, and we are very strict on dairy. All our commercial dairy people get rid of it, and it's as a result, because they tend to sell into the community, the level in the community of bovine TB is very, very much lower than human TB. That's it in a nutshell.
Thank you. And with that, thank you both for a wonderful presentation and the discussion. And we save the date. So Laura.
Yeah. Excellent. Thank you so much to Dr. Cheryl McCrindle and Dr. Rudovick Kazwala, as well as Dr. Tanya Bergerwolf for the excellent presentations and discussion, all highly relevant, very, very important. I would like to remind everyone to please join us for our 2020 Global One Health Conference. It will be Strengthening Global Resilience Through One Health, a 220 Vision for a Sustainable Future. And our registration link will be at go.osu.edu/register, GOHC 2020. And the date will be November 5th, 7th from 7 a.m. to 12:30 p.m. EST. So please join us for that. And thank you so much for joining us today. And we look forward to seeing you next month. Thank you so much.
Thank you. Thank you, everyone.
Thank you very much, and goodbye.
Everyone, appreciate it. Bye. See you, Rudovick.